All right, got this. I got this.
I thought that.
Hey guys. All right, so let's go ahead and get started. I guess before we actually get started, I also wanted to acknowledge we have some sponsors. So if you want your company name on our slide next time, it would be great if you give us some money. This helps us pay for the pizza and beer and so on. So it's always very appreciated. But without further ado, really excited to have Gina Nesr visiting us all the way from the other side of the United States, California. So she's currently a graduate student in Po-Soo Hwan's lab in Stanford. Before that, apparently was in Johns Hopkins doing some kind of SVD on protein sequence alignments and stuff like that. I think it's actually the first time I heard about you.
Oh, I haven't.
Because we were doing some related work. Yeah, so it's really excited to see now you moving into protein design space and now some kind of dynamics and so on. Yep. Trying to think of some fun fact. Let's see. Oh yes, that's right. So Gina also is the organizer of MLSB, and this time it's hosted in San Diego. Yep. Maybe I should just have you present that part. But the fun fact is weightlifting. So you guys need some tips on that.
This is such a great intro.
All right, I'll let you take over from here. Thank you so much for joining us.
Thanks, Sergey. This talk will not be about weightlifting, but very happy to talk about that after. I can probably talk about that longer than I can talk about protein dynamics. Okay, so I'm really excited to present, I guess this is the first half of my thesis work. That second half will hopefully be out sometime this next year, spring 2026. But for today, I'll be talking about learning millisecond protein dynamics from what is missing in NMR spectra. And this project was actually with Dorothy Kern and Hannah Wayman-Steele, who were actually both once upon a time in the Boston area. But I guess the prelude, I'm from Po-Soo Hwan's group at Stanford, and so we're a protein design group. And so you might be asking, why is a protein designer thinking about dynamics? And then why is a protein designer thinking about NMR? And then why is she thinking about it together? And so we've designed like thousands of de novo proteins. De novo proteins are, they're ubiquitous and they won the Nobel Prize in 2024. They've been very powerful at doing many things like targeting, sensing, and so on and so forth. And they're all fairly simple. But nature has evolved a lot of very, very specific and functional machines. They can do a lot of great chemistry, and they all use dynamics. So adenylate kinase has a multi-angstrom conformational change upon binding, which actually allows for its catalysis. GPCRs, which are a really important drug target, right? They also have a conformational change upon binding and actually changes the activation. Inactivation, cyclophilin A is an enzyme that has microsecond, millisecond dynamics, but on the residue scale, where there's 4 concerted motions of just the residues. And then myosin, being the machine of machines, converts the ATP to ADP, and in it actually has a full power stroke that takes 150 milliseconds. And so for us to actually do protein design that is on this grander and larger scale where we can do function, really the question comes down to how do we control this type of specificity and thereby dynamics. And so the central dogma of like sequence structure to function actually has this missing part of dynamics. And so if you think about it and how we think about the de novo protein design space, especially in the AI world now, it's really all fueled by the data sets that we have on each of these different realms. So in UniProt, we have over 2 billion protein sequences, and that's because every protein engineer has deposited the protein that they've discovered or learned about, every mutation in the information, so on and so forth. And so if you were to cluster that by 30% sequence identity, you actually get 360 million somewhat unique proteins. That's a lot of proteins in just sequence space. And that's what was used to fuel AlphaFold 2. With AlphaFold 2 also was the structure data. And so the structure data was experimentally characterized structures from cryo-EM, NMR, X-ray crystallography, and we have over 214,000 protein structures. That comes down to like 113,000 clusters according by like sequence identity. And that's a lot of really unique data for protein structure. And then even function, we have a lot of functional annotations, and people have gone in and taken their proteins and annotated, this is the residue that is responsible as being the base. This is the responsible residue for doing this function, for binding, and so on and so forth. And we have 250 million functional annotations. And so we have a lot of data to fuel a lot of AI measures. But really the question is what is the data that we have for doing protein dynamics? Because this is what is actually fueling a lot of that function. And so if we want to get to really specific, one way or one avenue of being able to do something like design myosin is to understand how we can actually transduce energy like this. And so if we think about what is really dynamics, it comes on multiple time scales. It goes from femtoseconds, which is almost instantaneous, to hours. So at the femtosecond time scale, we have bond vibrations, which is what you'll see like a lot of DFT and a lot of very simple methods looking at. And then at picosecond, you have methyl orientation. And so the methyl actually moves and transduces with it. At nanosecond timescales, you see a lot of local loop motions. Those are not necessarily concerted, they can just be random. It could be like the his tag that you use for protein purification, and it's just flopping around. But also a lot of other parts like IDRs and so on and so forth, also occurring in that nanosecond to microsecond timescale. And a lot of those timescales can be captured by molecular dynamics. OpenMM now is GPU enabled, and you can run molecular dynamics really fast. And it's what fueled actually some developments in things like BioEmu and AlphaFlow. And so we have molecular dynamics simulation that can capture this. But then all the really fun biologically interesting phenomena like enzyme catalysis and ligand binding and domain folding and so on and so forth actually occur from the low microsecond to like even the hour time scale in the natural protein space. And so we want to learn from that. We want to actually be able to capture that in terms of data and to actually learn what are the fundamental important values there so that we can actually use it for doing something like protein design. And so now if we think about what we have for dynamics, at the femtosecond to like nanosecond timescale, we have a lot of really great efforts to actually collect all that molecular dynamics data. We have MDCath, which is like the cath data set of non-redundant structures. We have a lot of molecular dynamics data sets there. Atlas has been a repository where people have deposited their molecular dynamics data. D.E. Shaw has done a lot of really long time trajectories at the microsecond timescale, and they've deposited, I think it's about 10 and a lot of shorter ones. What isn't at that time scale is the NMR ensembles and the PDV. Those are not indicators of dynamics. And so that's actually not an indicator of any of these things. But there are a lot of other repositories with that molecular dynamics data. But then you can go search for data on this microsecond, millisecond to like the second time scale, and you're not going to find anything. That's really sad because that's where all the fun biology happens. And so the question is like, how do we actually learn all of this biologically interesting and relevant protein dynamics for us to then go actually design and target and learn about biology at this time scale? And so NMR has a very, very strong observable, and we call that chemical exchange. Chemical exchange actually is a direct readout of microsecond, millisecond timescale dynamics. And so I'm not an NMR spectroscopist by any means, and I have happily in the last year learned a little bit, and I'll do my best to answer what I think is like the most important things about NMR and chemical exchange. So what you do is you take a protein and you'll label it. You'll label all of the atoms on it, the carbons and the nitrogens and the hydrogens, and then you'll put it in a big magnet, and then you'll get all of these really beautiful pixels here. You get little peaks. And then an NMR spectroscopist will spend a really, really long time trying to figure out which peak corresponds to which residue. And they'll do this painstakingly. The bigger the protein, the harder it gets. And then if they're actually interested in this time scale phenomena, they'll shove it into a magnet and run a different pulse sequence, and then they'll do relaxation dispersion. And so you get relaxation on two different time scales. This should say z-axis. You'll get R1s and you'll get R2s. And at the end of the day, you get information that directly reads off picosecond, nanosecond timescales, and you'll get REX, which tells you about microsecond, millisecond timescale. And so that's chemical exchange, is this REX value. And so this is important because this is how actually like biologists and biophysicists and structural biology study natural proteins to get this data. And they've done it on a large repertoire. But the unfortunate part is that this data is not deposited anywhere. And so what people will do is that they'll run this relaxation dispersion experiment, and they'll get some elevated REX component, which will look like this after you do the proper fitting. And so where there's something elevated, we typically will denote that in these little spectra as something orange. And if they couldn't assign the peak, it'll be red. We'll determine it as red. And they'll get this data, and then they'll go and say, what does that residue do for that function? And so Hannah, who initiated this project with Dorothy Kern during her postdoc, said, how many relaxation dispersion data sets can we actually find? And so she went and she scoured the literature and emailed a lot of people and was able to get her hands on 133 proteins, and then it became a little bit tedious. And so for all of these different data sets, we were able to actually identify every residue, what the dynamics label is. So is that residue likely experiencing picosecond, nanosecond motion? Is it experiencing microsecond, millisecond exchange? Or is there actually no data there? We couldn't see the peak on the spectra, or we weren't able to assign the peak on the spectra. Is it a proline? Proline don't have backbone nitrogens, and or it's very difficult to actually see it on the spectra unless you do very fancy NMR. And so is it a proline? There probably isn't data there, or were we just never able to get relaxation dispersion data for that residue for other reasons? And so we collected it for a variety of different proteins, as simple as GB3, the domain up here. Oh, there we go. To as complicated as something like beta-lactamase, which is interesting in its own biological right. And so we have 133 of them, and in the supplement of this manuscript, you can find all of them. And they've all been fitted and properly characterized, and we actually are able to identify each of those individual residues. So this is like incredible work by Hannah. And so we have all of this data, but if we want to see, is there something in there that could give us an indicator of an observable that we can try to use for training a model? 133 is not that big of a data set to train a model. I'm sure most people actually realize this if we compare it to something like UniProt. But if you take all of those residues and say, what are the things that we learn about here? We can just say, is there something relevant about the conservation of the protein? So if we build an MSA and we take for every residue, how conserved is it? And then correspond that with the label that there is. And so what we have noticed, and that was quite interesting to us, is that the residues that were indicative of microsecond, millisecond exchange that had some elevated, it had some REX in the R2 over R1, those were actually conserved. And there was a statistically significant conservation. Yeah.
For the 130 protein data set, are those all very different proteins?
Yeah.
And then mutant proteins, they're just proteins, so they're the same.
There's the de novo protein in there. There is some yeah, it's complete. They're very different. They're very unique, very separate from each other. Yeah, it's actually quite hard to find. I think a lot of relaxation dispersion set on the same like one person does it, and then they're like, we're gonna use that one unless we're interested in characterizing something very different there. But yeah, these are all quite different, and the data set's all public, and so someone can actually go and look at those labels for each of those individual proteins. Yeah. So if you were to take the sequence from RelaxDB and you were to build an MSA and say, how conserved is it through nature? So if we were to look at all of the sequences that people have deposited of related sequences, is there conservation on that or not? Yeah, just from an MSA, just from a naive MSA. Okay, yes. I'm going to talk about, if you wait 2 slides, I'll tell you what I will talk about. Yeah. Any other questions? Yeah. Yeah. So NOE is another like value that comes out of a relaxation dispersion experiment. And so you'll get R1s, you'll get R2s, and you'll get het NOEs. And so het NOEs, that value that is less than 0.65, we indicate as something having picosecond, nanosecond motion. Yeah. Just another value from an NMR experiment. Yeah. Okay. So build an MSA for each of those 133 sequences, identify how conserved it is by just basic conservation. And the thing we noticed is that the ones that are experiencing microsecond, millisecond dynamics that from the relaxation dispersion experiment had that elevated R2 over R1, they are also more conserved, especially in the core. And so we said, well, there's actually something that is missing. So residues that are exchange broadened, so sometimes the residue in the peak will have a nice little peak. If there's microsecond, millisecond exchange, it'll be a little shorter. But some residues can exchange completely and actually not show up on the spectra. And so those are peaks broadening. And so those don't actually show up on the HSQC. And so if you can't assign it, there won't be a deposition there, and then we can't actually evaluate an R2 over R1. We said, well, if the peak is broadened and it's missing an assignment, perhaps those missing assignments are actually experiencing or exchange broaden, and maybe those have something interesting there. And so we see are incredibly different, similar trends. So if we were to tabulate, oh, that residue is missing in each of those 133 data sets, that backbone amide wasn't detected and the NMR spectroscopist couldn't assign it. And then we build the same conservation and plot that, we see that the difference between those residues experiencing microsecond, millisecond dynamics and the residues that are missing is actually the same. So they're still also exchanged or also conserved. And so we actually said, well, what else do we see about this? I was really curious, maybe PLDT actually from an AlphaFold structure corresponded, but PLDT actually corresponds with picosecond, nanosecond dynamics. And so here the missing assignments might actually be something interesting that we can get out of NMR and we can actually use to train something down the road. We realize we actually have data on missing assignments for thousands of proteins. So we have 133 with full relaxation dispersion experiments, but we have chemical shift lists where an NMR spectroscopist assigned the spectra, they deposited it, and they actually were able to say, hey, we have these assignments, we couldn't find these other assignments. And so those ones, maybe those are also things that are exchange brought in, which are also experiencing microsecond, millisecond. The chemical shift lists that get deposited, some relaxation dispersion experiments, other types of experiments. And so we downloaded all of them in May 2023. So this was over a 2-year project. And there was 12,000 proteins at that time. And so we remove all of the different types of samples, the experiments that might actually dilute our efforts in this thing. We clean a bunch. There's actually no curation standard like the PDB for NMR data. And so we try to clean it, standardize it as much as we can, and we end up with 9,381 proteins that are in what we call this MBMRB. And so we can actually go in and say, did someone assign it? Is it missing, but it's like we don't consider it because it's a termini? Is it a proline, so we'll just ignore it? Or is it actually the assignment is missing? And so we have it for a wide range of protein lengths between like 10 to 400 residues. Yeah.
That's my question. I haven't done NMR, but the larger proteins are going to tumble slower.
Yes.
So are you losing any kind
We're just assuming very broadly that if it's missing, we just make that assumption, and we hope that the ML will like wash that, like will wash it away. The pre-training will wash that away. Well, the cutoff was actually in the training was 400 residues, and we don't have that many in our final training data set between 300 and 400. So the majority are actually less than, I think, 220. There aren't that many in there that are that long, but I think something really recently got deposited that was like 900 residues. I was like, this is crazy. Yeah. Do you have a question?
No, I was just going to say the distribution is kind of clear.
Yeah.
And you'll forgive me, it's mostly like 150.
Yeah, we did the data filtering after, and it, it reduces significantly when you remove redundancy. Yeah. Yes.
this later again, but how good is the assumption that missing residues are solely due to exchange products?
The complete assumption, we had a good amount of like predictive power on this, and that predictive power actually corresponds with other forms of NMR experiment, and I'll talk about all of that and the evidence we see there. We have a long ways to go. We definitely have a long ways to go, and this is just something that is an observable that we have on the orders of thousands. And so if we were to actually able to get data on larger order of magnitudes or of even better quality, so more resolved, I'm sure that we would be able to predict dynamics at a better scale. Yeah. Yes.
Kind of on the same thing, could you somehow correlate the dispersion of the spectra? Because sometimes, like,
If we had the spectra.
well, yeah,
We don't have the spectra.
if you have the range, sometimes, you know, if you have a very narrow range, it might be harder to assign peaks. So the reason they're not assigned is due to ambiguity rather than broadening.
Well, so from the proteins that we removed if there was like long stretches of proteins, if over some fraction of the protein was not assigned, so stuff like this. To hopefully remove stuff is cases like this, but that's certainly like something in the assumptions that is in there. I don't know if whether or not taking those chemical shifts and plotting it and saying, well, these are too much, because people spend a lot of time and they'll label it with their carbon and they'll do multiple different field strength and experiments to actually do their best to assign it if it's something new. So yeah, it's an assumption. And hopefully the proof is in the pudding, as I think we like to say. Yeah.
back to this assumption, right? So the way that the chemical exchange works is that when it's in two different chemical environments, the peaks are at two different positions as it gets broadened out.
Yes.
But let's consider a hypothetical case where I have a beta strand that's doing absolutely nothing. It's absolutely stable. I have an alpha helix that's moving
Mhm.
out.
Mm-hmm.
The beta strand, which is doing absolutely nothing.
Maybe we should let Gina get through her talk a little bit.
Okay. Because that's the problem with that. It does,
So,
it
we can
does
talk
that.
again afterwards, yeah.
Sure.
I know what you're saying.
Yeah.
We can chat about it after. I think we talked about this a while back too as well. Yeah. Okay. So we make this assumption, assignments are missing, and we use that to then actually collect and curate that data. So what we wanted to say is there other data that is this missing data similar or different than other forms of data that we have? So missing assignments are actually distinct from other forms of heterogeneity in larger data sets. So if we were to look at crystal structures and look at the same crystal structures for these sequences, are they corresponding, cryo-EM or so on and so forth. And so we see actually very little overlap between the data that we have from NMR and actually other information from X-ray and cryo-EM. In particular, something to me that really stood out is just that we have one NMR structure and 0 X-ray or EM structures for a lot of these proteins in the BMRB. And so that means that there are also residues we never trained on structure data for. And they're because they just couldn't get crystallized because they're so dynamic. So there's also information very much right there to get through it. And so I think someone in the back had said this, how good is that assumption? And so, yeah, we turn to the fact that deep learning is very robust to label noise. I think actually the bottom work is from a group here at MIT, and top work is from a Korean group. And they've really shown for other forms of deep learning that deep learning is robust to massive label noise. So if there is other reasons that a residue is missing, hopefully it's the small enough percent that it gets kind of washed away in that in the training process. And so we leverage that. And to just frame what exactly we're saying is if we assume that residues are missing assignments, they are exchange broadened, can we train a classifier on this binary 0, 1 if a residue is missing? And would that have, if something is missing, would that have predictive power on chemical exchange or on microsecond, millisecond dynamics? And so I want to really emphasize that we're at 9,000 proteins, and then all of the other form of structural and sequence data that we have are in the 100,000s to millions of proteins. And so we're dealing with very small data. And so what we really are thinking about is what can we leverage here to get this kind of predictive power? We didn't know what we could do with 9,000. And so we said, okay, what if we shove all the data in there? And so we combine all of that data, we split it by 80% sequence similarity split, which don't ever publish a model at 80% sequence similarity split, but we didn't know what would happen. So we said, let's just shove all the data and see if there's something there. And then we split it into train validation and test set. And we said protein foundation methods have learned to incorporate sequence and or structural information to learn a protein representation. So maybe we can leverage some of this. So we built really simple baselines. The baselines tell us about the random. Then Ramith, who was under postback in Sergey's group, actually built this AF2 pair representation, which took the pair representation from AlphaFold to see if that would actually have some form of predictive power. And it already outdoes the simple baselines that we tried. And so we think back and we say, hey, well, these missing assignments and also chemical exchange has this increased conservation. And so maybe something that has learned MSA information or conservation would be informative on this. And so it's a thing that deep learners love to do is look at which different representations in a big language model have learned what kind of information. We said maybe there's something in the middle there between that sequence structure relationship in the big language model that might have done it. So we took each of the individual layers from ESM2 and we trained a classifier to then just predict whether an assignment would be missing or not. And we already outdo this purely structural based representation. And then I think I had finished this ESM2 task and then ESM3 had immediately shown up in a paper somewhere. And I said, hey, well, this one also has some structure information. So what if we do the best of both worlds? And so we took ESM3 and said, let's just establish this basic baseline of just sequence space and learn that. And so it does actually a little outperform ESM2 a bit, and that's most likely due to the scaling laws. But then if you incorporate a sequence and a structural information of the protein of interest and train it that way, we see much more gained information. And so what the best performance that we got was out of layer 22 from ESM3, which had a sequence and structural representation in the final model. This is based on AUPRC, which I'm happy to discuss after why we chose on AUPRC and not AUROC. I said never train with 80% and then publish that. And so we went back and we took the layer 22 and then we did all the ablations. And so 80%, 50%, 30%, and then different structural cutoffs. And so our final model is at the 30% sequence identity and 0.5 TM score. And I want to note really that this final model is trained on just 2,000 proteins. And so the model representation here is carrying a lot of weight. And then the other thing is that the performance didn't change if we shoved all the data into the model or not. And this means it's a hard problem, both because maybe the assumption isn't we need more data on that time regime, but maybe also the model representation isn't sufficient to be capturing this time scale. Yeah.
Question for clarification.
For sure.
The model that you're training, is it a logistic regression model using the embeddings from ESM3, or are you training, what is the actual?
So you would input the sequence, and if you wanted to a structure, and it would run through ESM3, and it would take those model weights, and those would be the initial weights for a transformer. And the transformer at the end will just predict probabilities.
I see. So the model is transformer model.
Yeah.
And your input is the embedding.
Correct. And so the embedding gets built through ESM2, and that's the initialization there. Yeah, so it's not fine tuning, it's pre-training. Yeah. Yeah, Ben.
Did you try simple machines?
Yeah, they're horrible.
They're all.
They were really bad. So, hold on. I will jump to a. Don't look at anything. I don't know how to move anything. So we train a ton of stuff. So just like naive amino acids, conservation, coverage, just MLP, normal mode analysis, stuff like that, like just amino acid representations, basic DSSP, combinations of all of those. And they all pretty much perform on par with each other, which is not great. It's about as random as it can probably get. Yeah.
Random embeddings, like say you think the AIM.
It does the same as all of these. I think the embeddings are actually meaningful in this case. And you see that also in the middle of all of those independent layers, right? So the beginning of a big neural network is essentially it's closer to a random embedding, and that's usually much lower in terms of predictive power. Yeah.
One question I have, so SOP2 versus ESM2 versus ESM3, I guess one of the biggest differences may be evolutionary information.
Mm-hmm.
Do you know how you guys tried ALPHA2 with like sequence alignment information? I guess what I'm wondering is
If we've tried the MSA representation,
yeah.
I haven't tried the MSA representation, mainly because building an MSA is very time consuming. But that's certainly something one can try.
Because I'm kind of curious if the benefit of adding ESM2 versus ESM3 might be leveraging additional evolutionary information.
I think they're good, pretty. Oh, why am I raising my own hand? I don't understand. Sorry guys, I don't know why it's not letting me. There we go. So the difference in the bars between this and this is roughly the same between ESM2 and ESM3 with just the sequence. The difference here comes from just the structural input. And so the structural input is also like relevant in this case, I think. Between ESM2 and ESM3, I think the difference is like 0.1 or something along those lines. Yeah. Okay. We train with an appropriate split. Please train with appropriate splits. Please. Okay, and so then we take it, and our evaluation really is on the RelaxDB that we curated that actually has this interesting information. But first we wanted to say, can we actually just predict the missing assignments? So if we took our test set, our naive test set with missing assignments and predicted that, how does that do? So this is like one of the best performers. It's this really small ribosomal protein, which is involved in something in the ribosome. I don't know what it does. And so the assignment there is missing, and the probability there, we represent it with like a putty. So the thicker the putty, the more probable it is. And so this here, if you were to build an AUROC, has 0.9 AUROC. And so this is quite convincing for us. And then when we went back and we looked at some other ones, so this is something that had actually a much lower AUROC, but we had some predictive power in this loop. And if you go back into the BMRB entry, there's actually information here that says we couldn't measure the R1 and R2 for it because there was exchange line broadening. And so these are missing here. And so we can go through and actually do this kind of evaluation and say, what are we picking up on? Are these probabilities relevant to something that wasn't captured? Is the missing assignment the only thing that gets captured here? But really what we wanted to know is like, how well does it do on RelaxedDB? Are we really capturing chemical exchange? And so the model does have predictive power for assigned peaks with R2 over R1, elevated R2 over R1. And so we see that where there's the orange dots, those are the ones that have that chemical exchange, and there's a lot of them that are above random. But if you remember, missing assignments are also exchange broadened, and if we incorporate that, that whole part goes up. What is underneath the random baseline? Why are those ones getting predicted poorly? And so we went back and we actually looked at every single one of those and said, what do they have in common? And so what we realized is that our model doesn't have predicted REX if it's attributed to buffer binding. So these are experiments where someone took the protein, did the NMR experiment in phosphate buffer, but phosphate is also the same group in RNA. And so that protein, which typically binds RNA or DNA, was interacting with the phosphate buffer. And so we actually, there is chemical exchange in that experiment, but this is not necessarily, it's not in its like APO form. And so our model actually can't really predict REX attributed to buffer binding, likely because a lot of the proteins that were also in our training data set were also done in buffer binding if they were relevant data. And so those are all of the proteins that were RNA binding or DNA binding, phosphate binding, and were also characterized in phosphate buffer. So that's the majority of that bottom underneath that random baseline. We also noticed that the model had the most predictive power in core helices and then conserved core residues. And so that is just of interest to people interested in some form of phylogeny. So this is what all of those some of the examples from the model. BPTI, and next to it is an DE Shaw 5 millisecond separate, like 5 millisecond time trajectory from MD. And you can see where there is chemical exchange, and there's also a large delta in RMSD. For BPCI, we have predictive power there. My favorite is the flavodoxin, which if you've looked at the GitHub, I also have it in the image there. And so that entire FMN binding site, those residues are missing. We have predicted exchange there, and we have experimental exchange there, and we also have elevated probability from the model. And so we also see that like it's predicting things that are predicting dynamics and where there's like ligand binding or an active site or some form of interesting biological phenomena. Then there's the big boys of NMR, and that's called CPMG. And so that's a little different, bigger experiment that people do for a select amount of proteins. And when we curated, we were able to find 10 full data sets. And so we said, what does that data look like? And so we have R2, but you can get REX if it goes up. And so there's two different forms of values here. There's this R2 and this REX that is unsuppressed. And so this is what these 10 proteins that we have, beta-lactamase, adenylate kinase, cyclophilin A, a little less predictive on this like harder experiment, but still predictive. We realized when we were going in that there was a lot of predictive power at residues that didn't have an R2 value. And that was actually most likely because there is unsuppressed R2. And so what that looks like when you look at the whole spectra is where there's R2, you'll see at like different field strengths, the dots will go up, but you'll see there are dots that are already up that stay up, that don't really like have that striated rainbow color. And so those are unsuppressed R2. And so if we incorporate those and say things that are at that field strength that are being unsuppressed are also dynamic, experiencing microsecond, millisecond exchange, are also, and we incorporate that, all of our probabilities in that case go up. And so where is it? Oh yeah. So you see the probabilities all go up there. And then very recently, and so this is somewhat new and is actually unpublished, we did a prospective experimental test. And so we took our test set and we said we wanted to do 2 experiments. One experiment on a protein with high amounts of probability, and one experiment on a protein with low amounts of probability and little conservation. And so our first one was on this chitinase, which binds chitin as a part of its enzymatic activity in the binding loop. And so in the center there where it's circled is the active site. And then in the orange where it's also circled is the chitin binding site. And when we do the NMR experiments, we see all in that region, there is elevated R2 and we actually get an AU ROC of 0.7. Similarly, we take this protein, it's a hypothetical protein from one of the genomic studies, and it has very little predicted dynamics from Dyna1. And the NMR relaxation CPMG experiment shows that there's like no dynamics happening there. And so we were very pleased to see the experimental data match up roughly with what the values we were seeing from our test set. And so I think someone asked earlier is like, is this a good assumption? And so this is the assumption we have. Yeah, and so this is a great project with Hannah and Doro. We're really excited. Everything is up on Hugging Face, the data, the code. And because it was built on ESM3, previously it was a more restrictive license, but they recently announced that they were going to be open sourcing everything. And so Dyna 1 will also be completely open source for commercial purposes too. And so we're really excited for that. There's a Hugging Face for Dyna 1. It runs on H200, so it's super, super fast. You just drag and drop. And so it's super intuitive and easy to use. And then I'm really, really grateful for my advisor who will let me go on a whole tangent on NMR in the middle of a protein design PhD. And all of the people who shared all of this relaxation data. And I think with that, I can happily take questions. Yeah.
Just to, I didn't warn you before that for people asking questions, it probably, since no one has a microphone, it's probably best for people on Zoom.
Oh, that's a
if
good
you can
point.
repeat it.
Yeah, yeah.
But I do have a question.
Yeah.
Which is, oh boy, oh yeah, my question was, what do you mean? Do you think, like, you mentioned other neural network architecture maybe be better embeddings?
Yeah.
What do you mean? There's lots of models being trained on molecular dynamics simulations or being able to predict ensembles,
Yeah.
for example. Have you looked into that? Is that what you're currently, or what Hannah is at least currently doing, is trying to see if those embeddings for models that are
Like what models?
dynamic,
Like what models?
like BioEmu or other models that understand that trained on MD trajectories,
Yeah.
which I think BioEmu is trained on some of that.
Yeah, it is.
Do those embeddings offer a better representation of your prediction task, or are they worse than ESM3 if you started to look at it? Or
Yeah.
is it just something you're interested in?
So I guess for Zoom people, the question is, have you looked at other embeddings and what about the models that have been trained with MD? Good summary? Great. So MD tells you on femtosecond to like nanosecond, maybe really slight microsecond. That is not the big conformational concerted motions that typically happen between microsecond and millisecond. I wouldn't expect models that have been trained on these like small local loop motions to actually extend to it. And so BioEmu is a really interesting case, right? So if you go look in the paper, I think it's like figure 1A or figure 2A, they have the plot for adenylate kinase and the probability distribution, and they actually don't get the major state. So the state that adenylate kinase typically occupies is not predicted at all. And BioEmu is based on AlphaFold, right? So it's using the pre-trained weight from AlphaFold initially. So it's very interesting if we had maybe like longer MDs for more proteins, perhaps that's the avenue we could take. For sure, like still thinking about, I was thinking recently about like Prost embeddings and whether those would be more interesting. And I think there's a lot of different innovations coming out with different ML architecture and people conceptually putting them in. And so it'd be very interesting to see what other, like even just insights from what we get from this kind of analysis or data and then how that gets trained. Yeah. Sergey.
I guess maybe just to add a little bit to the answer. So the BioEmu is also using the AlphaFold 2 pair features. So I guess maybe one would expect that it's not just too clear what the second part of BioEmu is contributing over the pair features, but
yeah,
my guess would be that it might be equivalent to what Ramit was showing with the pair features.
Go to the pair features. I think the other thing is that like the pair representation is very like it's the PDBs that are like it's the structures that are in the PDBs. And so they're trained on very static structures, and a lot of the things that we're trying to predict haven't been captured by crystallography. I think Ellen Zong is a really good example of someone who's been trying to see what other information we can get out of cryo-EM. And then I think Stephanie Wankowicz also is interested in that in the X-ray crystallography. Hannah then being in NMR. So we've been covering our 3 structural biology spheres. Ben? I tried a really long list. I don't remember that list. I remember MLPs being in there. I think I really do think the model representation is holding a lot of ground there. And partly, like Sergey pointed that out, the conservation there. Oh, I forgot for Zoom. Ben is interested in why the transformer works, especially with 3000 proteins. So I think that the model representation there is holding a lot of weight. And because I think we've already learned some mapping between protein sequence and protein structure. And so layer 22 is actually like almost the dead middle of the model. And so it learns some intermediate representation between protein sequence and protein structure. And so maybe that is enough of uncertainty to.
Just to clarify, I just mean like the classifier layer that you're. Like
Yeah.
like
Oh, I see.
bootstrap.
Yeah. We tried like MLPs, we tried transformers. The transformer worked better because I think it had more weights to then actually try to compress everything in the protein representation.
Did you train the classifier?
Yes. Sorry.
A lot more CES data than CBH.
A lot more what? CEST.
Definitely, I think Google itself probably has more than 10 data sets, and probably I think about 50 to 100 CES data sets that you can have. That's give you other regional millisecond portion.
Yeah.
And not only
It'd be
that,
great to like grab that data and then see how we do on that. But hundreds is not enough to train a model. Yeah, but it's another, I think, so Hari's pointing out that CES data would be very interesting to account for here. CES data would be very interesting, and it's on the other time regime that we're maybe not capturing with just REX and exchange. And if we could get our hands on that and then see what those residues are, we'll get at least more information. Maybe not enough to train, but definitely enough to evaluate. And that would be awesome to standardize. Yeah.
talk a little bit about size of little data sets that talk about
From sequence or from structure or from NMR? Oh, okay. So we had what, 10,000, and then we filtered that down and we end up with 2,713 points, right? Yeah. 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 So those 2,700 proteins, I think what is really important to like note about them is that they're very unique. They're very distinct structurally and sequence-wise, function-wise. And so whatever those like future data sets look like, I think the most important thing is that they're very unique and diverse in sequence length, in functional annotation, in structural topology. What order of magnitude, I think, really depends on how well you can leverage the tools you have at hand. So no one would think you could train with 2,000 proteins, but we did. And so then the question is like, how well can you leverage all the other information that we have? Maybe we incorporate them into multiple heads or something like that.
Question about different kinds of data related to that. So have people also tried training, say, on residues that are missing in crystal structures that don't have any density, or residues with?
I haven't seen anyone do that. But the data that we have is very distinct from the data that's missing in X-ray or even like the data in cryo-EM. I'm certain that if someone were to go and tabulate that, we would get other interesting, important information from the X-ray data that we could incorporate. But I don't think anyone to this day has done that, to my knowledge. I'd love to be proven wrong.
How quickly do you improve?
Now.
There's a paper that I wrote.
Great. Sorry. There is a paper. I'm so sorry.
Fraser and myself, we did a lot of work on trying to understand could you connect motion in crystals with motion from spectroscopy, and there's a little equation that does it very nicely.
Oh, really? What's the name of the paper?
Yeah, you'll find it.
Okay. Find it. I'll write it down. Yeah.
Can you use your model to sort of come up with a list of, a top set of proteins that we don't already have NMR dynamics data on? Sort of a list for NMR spectroscopists.
Yeah. I have 9,000, and then we take the sequences that we don't have, and then we find the delta, and then we vote, I guess, as a community on which ones we want to know the most about ASAP. And then you find a grad student who's willing to assign all the peaks. That sounds about right.
You make mistakes in the assignment.
Yeah, I know. I'm aware. I've seen it. Yeah, if we had hands or we had more high throughput NMR, then there's a really long list. We only have 9,000.
Data in NMR train molecular data dynamics model. I've seen people use cryo, HTX. Is there a benefit to using full data sources?
100%. Is there a benefit to using other data sources? Yes, I think so. I think the main problem is that nothing is standardized, curated, and public. And so I think ours is standardized, curated, and public on this missing assignments and hopefully future data sets along these lines. The tough part about BMRB was actually just standardizing it. And so if we can get the standardization or even just like the identification and labeling for cryo-EM, for HDX, for all of this, that, hey, these are the observables that might be important here. I'm sure there's a lot someone could do with all that information. Yeah, we should all get together. You can get together at MLSP, come to San Diego. Okay. Yeah. Yeah.
Okay, if that's the case, did you also try fine-tune your domain by your own PSN?
I didn't. I think the compute was actually just beyond what I was able to do. With which ones? Oh yeah, you should be able to. I did not try that. I assumed that the proteins that ESM2 is trained on, because the repertoire was so large, it was gonna be representative enough to then just train something like that. But that's certainly something we could try.
How better off fine-tuning what's left in the layers than training a transformer from scratch?
Could be. I don't know. We'll just do we just do it. The day. There are membrane proteins in there. I don't know how represented they are. Harry, do you know how represented they are? Yeah. But they're definitely there. They're there. Yeah. They're definitely micelles. People use them. Yeah.
Maybe this is more of a clarification question. I think when you say the word transformer, people assume it's like a million parameter model.
Oh, it's very small, guys. This is a really tiny transformer.
Do you recall how many parameters of this tiny transformer roughly?
Parameters? No. It's 10 layers. It has 6 heads. So I don't know if anyone knows how to do the math for that. But it's to me, that is a fairly small transformer. Yeah.
I sort of have a philosophical question. Do you think, so, there's this structural genomics initiative. I don't know if that's still going on, but there is this, we're
Yeah.
trying to get as much extra crystal structures as possible proteins. Could your model?
That one was from the Structural Genomics Consortium.
Great. Could your model?
Just gotta say.
Could it apply, if you apply it to as many proteins as you want that are, let's say, 300 residues or less.
Yeah.
In all these sequence databases, could you rank and prioritize which ones we want to get really good? These ones are going to have really good dynamical data for us.
Yeah.
And let's just get the NMR spectroscopist like Hari some money to collect the data for the structural genomics like dynamics data sets that we need for training better models. Is that a crazy idea?
I don't think so. I think what you guys just need the money and people willing to do NMR, and maybe throughput and maybe more money. So
money, that's what we can do.
money, I think, people willing to do NMR? I don't know how hard that is. It's not crazy, but if you find the person who's willing to do it, or we develop new techniques to do that. Yeah.
Last question for me. I think a few people mentioned HDX.
Yeah.
How different or similar is that NMR, and is that more scalable?
It's a different experiment. Yeah, what would be the difference between NMR and HGX for data set creation?
I mean, they're different, but I mean, but would they capture similar information about what's missing? Like
I don't think I'm familiar enough with HGX to properly comment on that, but I would imagine that they would tell on different stories about the same protein, and it would be different informative stories.
looks like we have an audience member that is.
So if the audience member who knows,
Protein, it was one of the standards, right? So it was in, things like this. So he was 2 years into it, and parts of the protocol still not changed, right? Right? And the challenge with microseconds to milliseconds is that you can't easily do an HT exchange experiment by mass spec, if that's what we're talking about, using those techniques. Now, NMR spectroscopy has great ways of doing it, right? It's deuterium hydrogen exchange. You look to see whether the proton exchanges are, right? So we can do those kinds of experiments. Really, this microsecond to millisecond time scale is it's really been a lot of techniques, a lot of physical techniques. So standard kind of like let's find out what's kind of solvent exposed. It's not that right? I'm afraid it's people. It's not your oracle.
Yeah, thank you so much.
better answer than I would have provided. Thank you.
So I'm really curious about your curation of the HSQC data sets. Because as you rightly pointed out, there's a lot of emissions and alignments.
Mm-hmm.
So you can't have a single amino acid that's dynamic. So there's probably near the missing dynamic, the line with all these starts increasing.
Mhm.
Is that probably somewhere you guys are trying to leverage that?
I'll tell you about it on Friday.
Okay.
Cool.
Still got 6 more minutes for any.
So the whole 6 minutes. Are there any on the Zoom? I don't see any. I remembered. Are there any questions?
So I have a question too.
Yeah,
You did the last layer, who's that?
22nd.
No, for ESM3 you did.
Oh, for ESM2, yes, the last layer.
What, like, was nothing better than the last layer of ESM2?
No. Well, we went through all of them.
Yeah, but it was.
Yeah, it was useful. I did ESM2 and I did ESM3 and I was like, am I wasting my time into my compute? And then I did ESM3 with a structure and then was glad that I did this exercise.
I'm seeing a lot of companies today.
Yeah.
I think we're trying to understand from the perspective of what's happening with the CZ, what we can use.
Yeah.
I'm open.
So I'll tell you my insider scoop from another group that is using ESM3, myself who has used ESM3, and then EvoScale who built ESM3 is that layer 22 of the 650 million parameter model is the layer to use, I guess. But then they also have the scaled ones that are open, and I think those will be very fun to play with just because of how big they are.
Yeah.
Yeah.
Right. We often see people like the 3 billion parameter model, but then there's always.
There's the 7 billion one, which I've been told is I should go play with it.
It keeps giving.
Yeah.
Keeps it just going.
Yeah, there's some paper that came out with like scaling of language models, and there's how good their performance is on like protein gym, and they show a plateau. It does end up plateauing at some point. So really, we just do need better representations.
So I have another question about, so you mentioned data splitting,
Yeah.
right? So here's my question about data splitting for this problem.
Mm-hmm.
Is, you know, if you split by sequence identity, which is the very common way to split data, of course, there are certain parts of proteins that can be very conserved when other parts of the same protein is very divergent. And I use the example of binding pockets for this.
Yeah.
And so you can fool yourself thinking that you have a dissimilar protein in the overall sequence, but if the task is binding pocket prediction, you might have identical binding pockets in your training task. So my question, that's my background.
Yeah.
My question to you is is there something really worth considering for this particular problem in terms of, weird data leakage that you might find? Is there like a particular loop? If you have this loop in your protein, you will have this dynamics, and you really need to make sure you filter out for contiguous sequences that are dynamic and known to be dynamic. And in order for like really understand or knowing that you don't have trait test leakage. Like, I'm just wondering, are there
that's really interesting. I haven't thought about that. Well, dynamics isn't just like the very local part that's the pocket, but everything else that's supporting it and facilitating that function, right? So you have the same residues that maybe like I can recognize and bind ATP, but you also it's being facilitated in different ways with respect to the rest of the protein. So for the case of adenylyl kinase, it's the entire domain fold, the closing. Whereas with the power stroke in myosin, it's still the same residues that are recognizing it, but in a different configuration or in a different mechanism to allow for a completely different amount of function. And so a lot of the exchange that gets recognized, yes, it might be like that binding pocket, but it might be also the residues that are anchoring and allowing for the entire domain movement. So I think a very conceptually and probably a fun rabbit hole to dive down. But also sounds quite difficult in terms of like identifying like directly identical pockets. And then if you go down the route of like how similar are these two loops, you might not end up with any data. So I think it's a fun, interesting thought process. It's a fun, interesting thought experiment.
Yeah, but all of the data, the indexation data were measured at the same temperature.
Excuse me?
Was everything measured at the same temperature?
No, they weren't.
Of course.
Yeah.
So there might be possibly the ability to tune what looks good in the spectra based on the temperature that makes things look good. Right? So just be aware of that.
Well, let's thank Gina again.